Hi, my name is Chanel, and my other group members are Sammy, Xavier, and Jenna. Our group decided to research the symptoms, transmission, controlling transmission, and treatment of yellow fever. Next slide. So I focused on what yellow fever is and its symptoms. Next slide. So yellow fever is an enveloped RNA virus that is transmitted by mosquitoes. Its origination is from the tropical and subtropical parts of Africa and South America. Once transmission occurs, the virus exists in just the blood of the host, which is called the period of infection phase. The virus replicates in the lymph nodes and infects dendritic cells of the body. It then spreads to the liver after replication, which then triggers cytokine release syndrome to occur. Cytokine release syndrome is the body's systemic inflammatory response to an infection. It causes the release of large numbers of white blood cells, which then release inflammatory cytokines to fight the infection. If the cytokine release doesn't help fight the virus off enough, shock can occur, which causes insufficient blood flow to the tissues of the body, thus often leading to death. This immune response to virus creates the unpleasant symptoms of yellow fever that I'll go over in the next slide. There we go. So when somebody becomes infected with yellow fever, there's an incubation period of 3 to 6 days, where there are no symptoms. After the incubation period, symptoms start to take effect quickly. One of the symptoms includes a fever that can range from 102 to 105 degrees Fahrenheit and last several days and can cause heart rate to slow. If the host recovers from yellow fever, they can go into what's called the period of remission. If the host continues to be infected, they will experience headaches, dizziness, nausea, muscle aches, loss of appetite, and lastly, a red face, eyes, and tongue. Next slide. If the subject does not go into the period of remission phase, they will then go into what's called the period of intoxication. Approximately 1 in 7 people that become infected with yellow fever get to this phase. This is where the virus disappears from the blood and starts replicating in the liver, and antibodies appear in the body. When replication comes, symptoms of brain dysfunction, bleeding, abdominal pain, bradycardia, which is the slowing of the heart, decreased urination, kidney and liver failure, and lastly, jaundice, which is yellowing of the skin. Next slide. The critical phase of the illness occurs between the 5th and the 10th day, at which point the patient either dies or recovers rapidly. Symptoms of the critical phase include hypotension, shock, hemorrhage, metabolic acidity, and sometimes death. Death rates are the highest in infants and elderly, and death rates have a ridiculously high variation because of different reporting methods in different areas. So we're not really sure of like a solid death rate. Next slide.
All right, I'm Sammy, and I'm going to talk about the transmission of the yellow fever virus on the next slide. All right, so as previously mentioned, the yellow fever virus is a flavivirus that originated in Africa. The disease, which was originally thought to be passed from person to person, which is not the case. Yellow fever is spread when an arboreal mosquito, typically the species Aedes aegypti, bites a human or non-human primate and infects them. The picture demonstrates the transmission cycles of how the disease is spread by mosquitoes. The urban cycle was the first cycle to be discovered in the 1700s, when a large number of infections began appearing in the port cities. In the urban cycle, transmission occurs between mosquitoes and humans. The virus is brought into urban areas by a viremic human who became infected after spending time in the jungle. The sylvatic cycle was the second cycle to be discovered after finding NHPs or non-human primates that possessed the yellow fever virus. In this cycle, the virus is transmitted from infected mosquitoes to monkeys, then to the humans that enter into the jungle. The intermediate cycle occurs only in Africa, and the virus is transferred from monkey to human or human to monkey from the infected mosquitoes. Spillback between urban and sylvatic cycles had been a concern in the spread of yellow fever virus. Spillback occurs when a reservoir population that possesses a high pathogen prevalence comes into contact with a unique host population. One study I looked at investigated the likelihood of a spillback occurring in Brazil as urbanization increases. They collected a total of 5,000 mosquitoes from urban and jungle locations and 150 non-human primates before and during a yellow virus outbreak in Brazil. The mosquitoes tested in urban areas were all negative for yellow fever virus, while only some of the mosquitoes acquired from the jungle possessed the virus. The virus also quickly and fatally overcame many of the test subjects, preventing mosquitoes from acquiring and spreading the virus. These tests suggest that the disease was not caused by spillover and was actually caused by the sylvatic cycle, indicating that spillback or spillover was not likely an issue in spreading the disease. And next slide. All right, and there have been other means of spreading the disease that have been considered as well. It was suggested that males infected with the yellow fever virus could pass it to a female and her oocytes during mating. There is also some concern that infected female mosquitoes are capable of passing along the yellow fever virus to their offspring by means of vertical transmission. This causes concern because vertical transmission is capable of maintaining a virus when conditions are adverse. In a study I found from the American Society of Tropical Medicine, they collected 100 female mosquitoes of varying 80 subspecies and infected them with a flavivirus, similar to yellow fever. These females bred the F1 offspring in the lab where scientists collected and hatched the larvae that the females had produced. They found that some of the F1 offspring were infected with the virus, but infection rate differed among the subspecies. However, they found that the virus could only survive a short time without horizontal gene transfer occurring and would not be passed onto the F2 generations or any subsequent generations. Determining how the virus travels can prove beneficial in tracking where the virus will likely spread and to maybe even one day help us develop a cure to treat it. And this is just a chart showing the ratio of the infected offspring. And next slide.
Awesome. I'm Xavier, as you can see by my face. My subsection focuses on the treatment of yellow fever. Next slide. So there is no cure for the viral infection of yellow fever. The only treatment accessible today treats or manages the symptoms of yellow fever. Current research has focused on antiviral medication testing to treat the viral infection, as well as preventing the viral spread within an individual to prevent the treatment of symptoms. A yellow fever vaccine is readily available, though, and can prevent a majority of future treatment. Next slide. Antiviral medication. Current research has been testing numerous antiviral medications' effectiveness of eliminating yellow fever viral infections. Such medications include ribavirin, teizofurin, carboxamide, and pyrazoline. These medications have been tested on numerous mammals. Out of 4 antivirals tested, ribavirin was the only one with some success of treating the viral infection. Ribavirin cured the viral infection in hamsters up to 2 days after the initial infection. This is the time span before the infection reaches the liver. Although there was 0 success of infection removal in non-human primates. Regardless of this lack of success in primates, there is still potential for a combination of ribavirin and synergistic drugs to successfully treat yellow fever viral infections. Next slide. Limiting infectivity in an individual. Fady et al. 2019 performed a study focusing on the treatments that would affect the infectivity of the yellow fever virus in human plasma cells and platelet concentrates. 2 treatments were conducted. Teraflex MB was a visible light treatment in the presence of methylene blue and was conducted on plasma cells. Likewise, Teraflex UV was an ultraviolet C light treatment and was performed on platelet concentrates. The results of the treatments found Teraflex MB successfully reduced yellow fever virus infectivity by 4.77 log in plasma cells. Furthermore, Teraflex UV reduced infectivity by 4.8 log in platelet concentrates. The implications of these results are both treatments can effectively reduce yellow fever virus infectivity within an individual. Additionally, these treatments could be used to manage the transmission and transfusion within an individual's plasma cells and platelet concentrates. This can help reduce the spending and use of resources put towards the treatment of symptoms due to the 2 treatments' ability to reduce viral spreading. Although near future wide-scale use is likely to be difficult due to the lack of funding and resources where the yellow fever is prominent, which is South America and Africa. Next slide. Preventing treatment. For a long time, yellow fever vaccines have been developed and used. Though they've been used for decades, their effectiveness went relatively unknown for some time. Beginning in 2009, testing of the yellow fever vaccine was performed on 318 participants in Brazil. From then to 2017, these participants were monitored annually to test for vaccine seropositivity. Through the 8 years of monitoring, 85% of non-revaccinated individuals remained seropositive for the vaccine, although the highest success occurred in individuals with vaccinations of 587 IU or greater. These results prove the efficiency of the vaccine and have aided in the prevention of future treatments across the world. There is promise to find a cure for yellow fever, but until then, finding a way to treat the virus internally and preventing treatment altogether is all can be done. Next slide.
All right, so since there is no way to cure the virus once it's in or once a person is infected, and although there is the vaccine, not everyone has access to it. A lot of the people in the areas that it's endemic are poor and maybe don't have access to vaccines like this. So one of the ways to control transmission is to target the Aedes aegypti mosquito, which is the vector for this virus. And one of these methods involves the use of insecticides to control the mosquito population. However, this isn't the most effective technique because you would have to reach all of the mosquito population, which can be hard to get as they are widespread and they can be either indoors or outdoors, and they can develop resistance to the vaccine or the insecticides pretty easily and quickly. And insecticides are also bad for the environment as they can affect non-target organisms, and they can, accumulate in animal tissues over a long period of time, which includes birds and mammals, including humans. So even humans can be affected by this. So they're not greatly, or this technique isn't really super effective, and it does have negative impacts. Another technique involves kind of the same idea, lowering the mosquito population, but it's a lot different of a technique. It involves genetically modifying mosquitoes to have a lethality gene. And this gene, they put in the OX513A mosquito, and it makes mosquitoes require an antidote called tetracycline while they're developing. And in the lab, they can feed the antidote to the mosquitoes and grow up the populations, and then once they're released into the wild, the mosquitoes, once they breed, all of their offspring don't survive development because tetracycline is not available in the wild. In this figure from this study, the dotted line shows the density of mosquito population in areas that weren't treated, which, as you can see, the population stays pretty steady. However, in areas, if you can see, the solid line, that shows the mosquito population in areas where their treated mosquitoes were released. So this technique can cut wild mosquito populations around 80 to 95%, which is pretty huge. And it doesn't have as bad of environmental impacts because it's not targeting other organisms that aren't the mosquitoes, obviously. However, it removes, since mosquitoes are a pollinator, it removes the pollinator from the environment, which could affect biodiversity in these environments. There's one more technique that I looked at, called the antipathogen gene. So it involves the same genetically modified mosquito, well, a different mosquito with this gene in it. So this gene is called defensin A, and it's added to a regulatory region of the genome, which is activated after a blood meal. So basically after the mosquito eats blood or has the blood meal from the human or other animal, it activates this part of the genome, which correctly processes into a mature defensin protein. And this protein basically increases the immunity of the mosquito to certain bacterias or viruses or pathogens. So in this case, this gene produces antimicrobial characteristics when infected with a certain bacteria. However, it has promising implications as if scientists can improve the technology that they use and test this gene a little bit more. It's possible that this gene can be used to prevent the mosquitoes from getting other diseases like the yellow fever virus, so it's still in the testing process. However, it's very promising, and it could turn into something great in the future. If the mosquitoes don't get the virus, then they can't pass it on to us. So it could be really effective in preventing transmission. And I believe that is all we have. So thank you for listening to our presentation. Does anyone else have anything they want to add?
It was great.
Okay. Let's see how to stop it.